Hi, in this video we'll talk about oxidative phosphorylation, which is the main method that cells use to make the majority of their ATP. We'll talk about the mechanism that ATP is made, which involves the coupled vectorial processes of electron transport, establishment of a proton gradient, and the use of that proton gradient to drive ATP synthesis. And finally, in this video, we'll look in detail at the structure of the mitochondrial inner membrane, which is the location for most of these processes, as it is the essential barrier between the inner mitochondrial matrix and the mitochondrial intermembrane space. Now, in our prior lectures, we saw the process of glycolysis, where cells took glucose and other carbohydrates and managed to obtain a small amount of ATP using substrate-level phosphorylation in an oxygen-independent manner. Today we're going to talk about the much more efficient mechanism of generating ATP, which utilizes oxygen, and that involves capturing high-energy electrons in the carrier molecules of NADH and FADH2, which then shuttles the electrons to the electron transport chain. Just to review, in glycolysis we took glucose, converted it to pyruvate. In the process we generated a net of 2 ATP and 2 NADH molecules. In the citric acid cycle, for each rotation we generate 3 NADH molecules, a molecule of FADH2 and a molecule of GTP, and we do that twice because glucose produces 2 molecules of pyruvate. We'll see a more detailed accounting in future slides. And this is just a reminder that both glycolysis and sugars will create pyruvate, which will funnel into acetyl-CoA, and fatty acids that are imported into the cell get transported to the inner mitochondrial matrix, undergo beta oxidation, and produce 2 carbon units of acetyl-CoA as well, allowing both acetyl-CoA types of molecules to enter into the citric acid cycle. Today's lecture will focus specifically on the process of NADH donating high-energy electrons to an electron transport chain, which will drive the formation of a proton gradient, which is then used to drive the synthesis of ATP, and the electrons will eventually get dumped onto oxygen, forming water as a byproduct. In addition to talking about oxidative phosphorylation, or the process of respiration that animals employ, we'll also in later lectures look at photosynthesis, which is the mechanism whereby plants and photosynthetic bacteria and algae produce ATP. The two processes share a lot of features which are highlighted in this slide. Both utilize high energy electrons. In photosynthesis, the electrons gain their high energy from the absorption of a photon, the energy that's present in sunlight, and in foodstuffs it's from energy-rich molecules such as carbohydrates. In both cases, the high-energy electrons are used to pump protons across an inner membrane, creating an electrochemical gradient of protons, which can then be used primarily to drive ATP synthesis, but can be used for other processes in some organisms as well. The term chemiosmotic coupled processes denotes the fact that electrochemical gradients across a membrane are going to be used to drive a particular chemical event, which in this case is the synthesis of ATP. And here we start to see some details of chemiosmotic coupling. High energy electrons will be donated to a protein. The fact that the protein now has an additional negative charge will change the conformation of the protein. This conformational change will result in pumping protons from the inner mitochondrial matrix to the inner membrane space. This creates an excess of protons here relative to the inside of the membrane. And then after the proton pumping has occurred, That will again change the conformation of the protein. Now the affinity for that electron is decreased, and this electron will be passed on to another electron acceptor, whether it be protein or small molecule. Now in the second stage, we use the excess protons that are available in the inner membrane space. They will diffuse down their electrochemical gradient in an energetically favorable direction, and that energy will be captured partially by the synthesis or the coupling of inorganic phosphate and ADP into the energy currency of the cell, which is the molecule ATP. This process of osmosis and chemistry forms the fundamental basics of chemiosmotic coupling. Now in this slide we start to schematize some of the events of oxidative phosphorylation. As we know, acetyl-CoA that's produced from fats and other high energy molecules enters into the citric acid cycle. The carbon atoms of acetyl-CoA will finally get excreted as carbon dioxide, which is as oxidized as a carbon can get in life forms, and there's no more extractable energy from that. As it's being oxidized, high energy electrons are being captured on NADH, which then will diffuse over to the inner mitochondrial membrane and donate those two high energy electrons to a series of 1, 2, 3 different protein complexes that all reside within the inner mitochondrial membrane. Now these are schematized here to suggest that these are the highest energy electrons, these are medium energy electrons, and these are lower energy electrons. Each time an electron is placed on this protein complex, it will pump hydrogen ions or protons, that's the same, synonymous, from the inner mitochondrial matrix to the intermembranous space out here. After doing that, the electrons will get transferred from this protein complex to this protein complex, which will again pump protons, and again at a third protein complex. At this point, the electrons are of low enough energy that they can no longer sufficiently drive protons being pumped, and so they are dumped onto molecular oxygen that we breathe, and it's going to pick up some protons from the internal milieu, and the electrons plus oxygen plus protons will form water as a byproduct. And again, this is exactly what happens when you burn hydrocarbons. You produce carbon dioxide and water. On this slide, it's just a reminder of how NADH carries these high energy electrons as part of a hydride ion, which then when it approaches the protein complex will donate 2 electrons to the electron transport chain, a proton to the internal milieu, and this will reform NAD+, which is the acceptor molecule for the electrons in the citric acid cycle. This is once again the electron micrograph of the mitochondria, which we saw in the first class. Just as a reminder, there are 2 membranes to this organelle. There's the outer membrane and the inner membrane, which folds in here. And so there's 2 spaces. There's the inner mitochondrial space, which is the inner mitochondrial matrix, which is the bulk of the volume here. And then there's a small space between the outer and inner membrane called the inner membrane space. And this is the location which protons will get pumped and will achieve high concentrations in the small space. The proteins in the inner mitochondrial membrane will now be pumping protons from the inner mitochondrial matrix into this intermembranous space, the small space, and so even a small number of protons will greatly increase the concentration, and then the protons, the excess protons in this space, will then be transported back out into the mitochondrial matrix, driving the synthesis of ATP. So one of the key advantages of having these 2 membranes is the creation of 2 separate compartments, the inner mitochondrial matrix and the intermembranous space, in which different chemistries can occur, different concentrations of molecules can be maintained, and the differences can be used to drive important processes. And this is a good place to stop. We'll resume this video with a discussion on the events happening at the inner mitochondrial membrane. Thanks for listening.